Can you hear me now?
Yes, we are live.
Okay. I can go ahead and talk.
Yes.
All right. Well, hello everyone. My name is Dr. Chung Lee. I'm the professor and the chair in the Department of Neurology at Houston Methodist Hospital. Um, it is my pleasure to meet and talk with you about CMT type 4. I want to just briefly explain about what is the CMT type 4. And here is a cell that every human being every cell have the genetic material called chromosome that carry the genetic information that inherit from mom and dad to the next generation. And then on the chromosome, there's two copies for each individual gene. If a disease caused by mutation in one of the copy of the gene, it will call out some dominant type of the disease. But if the disease only caused by mutation in both copies, then we call out some recessive disease. CMT is CMT type 4 are the inherited peripheral nerve diseases caused by some recessive mutations, means the mutation has to occur in both copies, not just one. So that's the definition of CMT type 4. And then when the patient who have CMT type 4 typically have the following symptoms or physical findings. They could have a sensory loss in their feet, leg, hands, or arm. They could have pain, pins and needles, and all other altered sensory symptoms. The disease also often affect the muscle, so the muscle will be weak and appear wasted, and so therefore the volume of muscle will smaller than the normal human being. On physical findings, when the neurologist examine the patient, they often find, in addition to the sensory loss, muscle weakness, wasting, they also find there is a decrease of deep tendon reflexes. They also find other abnormalities in the non-neuron neurological organs or tissues. For instance, there are some CMT type 4 could affect the spleen or liver, but again, the major portion of the symptoms are derived from the nerve system. So, in order to understand how the therapy has been developed for the CMT type 4, I would like to introduce a concept of loss of function mutation. So here is the diagram show a gene. The gene gets transcribed to mRNA, and after mRNA formed, these will engage with a translational machinery and read the codon from mRNA and eventually produce a protein. In the situation of CMT type 4, mutation affect both copies of the gene and typically will either eliminate the translation of the protein, means that you don't have protein at all, or there's a significant reduction of the protein being made. So we call loss of function mutation. So for that reason, that CMT4 typically treat by the following modalities. The first category, what we call symptomatic treatment, means that if the patient has pain, you will give the medication to control the pain, or if you have weakness in the ankle, may treat it with ankle braces, and all these symptomatic treatment aimed to improve the quality of life. With the advance of molecular genetics, now there's a rapid evolution of gene therapy. The concept of the gene therapy is to replace the loss of that gene product. We just introduced the loss of function of the gene mutation in the CMT type 4. So therefore, it would make sense if you bring that gene product back to the human body. And so the way to do that is either deliver that gene through the viral vector, like here, shown here, or through the system that not viral vectors, but such as nanoparticles or other vehicles bring these gene products into the human cell. Alternatively, you can take some cells out of the human being and then correct that gene mutation and then transplant the cell back to the human body. So there's several ways to reintroduce the gene back to the human body and in order to treat the disease. CMT type 4 in this case is a loss of function. So that is a brief discussion about the CMT type 4. And I'm going to pause in here, and I'm happy to answer any questions from the audience. And I'll stop sharing here now.
Alrighty, thank you so much, Dr. Lee. I'm just waiting to see if we have any questions coming in. It looks like we have one new question. Any new developments for 4J?
Yes, so the CMT4J is caused by, just for those in the audience who are not familiar with CMT4J. So CMT4J is caused by some recessive mutation involve a gene called FIG4, spelled F-I-G, number 4, FIG4. When that happened, and then the human cells either express the protein FIG4 minimally or not at all. In that situation, the patient will have severe demyelination of the Schwann cells as well as the neuronal degeneration. So the patient will have weakness, sensory loss, muscle wasting, sometimes could affect the respiratory muscles, so therefore difficulty for breathing. And there are individual patient may also suffer from the spleen problems. So all these symptoms are caused by what we call loss of function of the FIG4 gene. So there have been animal model made for this specific disease, and then this disease is suitable for gene therapy, means that bring the gene back to the human cell. So there have been a publication where they use gene therapy and the viral vector bring the gene back to the mouse model, which showed a significant improvement of neuronal degeneration and also other deficits that see in the animal model. There have been efforts to try to translate this to the clinical setting, and we under the FDA advice, there is effort to do the natural history study of CMT4J, and that data is still pending, and the natural history study it will be very important before the actual gene therapy started, because that's the baseline information for this disease. And so in summary, the progress has been made, has a successful in the animal model, but have not get to the human yet. So that's the that would be my brief answer for this question.
Awesome. Thank you. So let me bring up the next question. We'll show it on stage. Can you speak to specific treatments for 4C?
Yeah, so specific 4C is less familiar to me. It means that the therapeutic development, but I will briefly introduce what I have known about this. And the CMT4C is caused by a mutation in a gene called SH3TC2, and these patients usually have a early onset spine deformities and weakness in the muscle, and also the high arch feet, hematose, as well as some sensory loss among these patients. I personally have seen a few family, or by the way, that these patients also have a hearing loss, vocal cord involvement, and facial weakness. If you do the EMG nerve conduction study, they will have slowed conduction velocity, and those are the common symptoms among these patients. So there have been the research ongoing in the academic centers where people have identified a mechanism of this disease and find a specific small molecule compounds could act on the pathogenic molecular pathway of this disease, and it has reached some degree of success in the animal model, but I have not seen these have successfully translated to the clinical setting yet.
Alrighty, so let's go to the next one. And it looks like you might have already just spoke on this, but we'll go over it. I saw 4C listed as a second only to type 1 developed successful animal gene therapy. How familiar are you with these studies with Professor Cleopas?
The study for Dr. Cleopas, I heard from his presentation. It basically uses a gene therapy, a viral vector, bring the gene back to the peripheral nerve, approach similar to what I described for CMT4J. It has achieved some degree of success in animal model, but I have not seen these have been fully translated to the clinical setting yet.
And then likewise, is there any currently clinical trial with gene therapy for 4C?
I am not sure because not I knows of at this point, but I could miss somewhere. As far as to my best knowledge, I have not seen a current ongoing CMT4C trial.
Okay. Let's see here. All right. It looks like all of our questions in our Q&A have been finished. Just want to go through the chat. anyone has any other questions for Dr. Lee, please put them in the Q&A and we can ask him. We've just got a few more minutes before this session concludes. Looks like one just came in. Any updates on 4A?
4A, let me just briefly introduce about 4A. And 4A related to a gene called the GDAP1, and spelled GDAP1. And this gene is involved in the mitochondrial regulation. And a few years ago, my lab also have studied a few family with the mutation on this gene. There actually have lots of research ongoing for understand the biology of this gene. And in terms of the therapeutics wise or gene therapy wise, and I have not seen a direct translation to the clinical practice yet. And so that will be my brief response for this.
Alrighty. Any cross medications that could show promise?
To which disease, which one, or is generic? Is this general?
I think just general, yeah. Or it looks like potentially 4J.
Oh, the 4J. I have seen quite a bit of the 4J patients personally, and among some of the 4J patients, they have a significant segmental demyelination on their nerve. And I have seen some of the patients, at least initially, they get some response. And again, this is not controlled clinical trial and based on individual patient. They seem to initially respond to the intravenous immunoglobulin, presumably that help for that demyelinating condition. And they improve to some degree, but then after a while, the effectiveness declines. Again, that presumably due to the continued neuronal loss, axon loss. So this effect is not long lasting. And that's the observation that I had in the past. And other than that, I have not seen any other medication has been made to the clinical practice yet.
Okay, we've got one last one. You mentioned that one molecule might have positive effect on CMT4C in animal models. Do you know if the corresponding study has already been published and is available on PubMed?
That's a good question. I heard the investigator present in the meeting. So I have to check if that small molecule compounds has been published in the PubMed. I'm not sure.
Alrighty, so that looks like we are out of questions at the time. Thank you, Dr. Lee, for your wonderful and informative presentation. My name is Sarah Gentry, and I am the Technology Manager for the CMTA. I wanted to take a minute to say thank you to everyone, including Dr. Lee, who has joined us today for a full day of learning and connection. We truly appreciate your support, and we plan on having all of the session recordings available in the coming weeks. If you have any questions, please feel free to reach out to info@cmtausa.org. Thanks so much, everyone. And this now concludes our 2022 Patient and Research Summit. Have a wonderful rest of your day.
Thank you. Bye-bye.
Thank you, everyone.